Good morning, everyone. On the second day of our Big Data in HPS conference, after a very productive first day, it is my pleasure to present our keynote that opens this morning session, Dr. Charles Pence. And he has done work in history and philosophy of biology, especially on historical conceptions of fitness and chance in evolution. Is published on issues pertaining to the use of deep learning algorithms on historical scientific text, big data approaches in digital humanities, and was part of the Evo text developing. So it's my pleasure to introduce Charles Pence. Charles, take it from here.
Yeah, thanks so much. This is the first day was already really fantastic, so I'm super excited to get to be here and share a little bit of this. Yeah, just to tag on there. Yes, you don't know who I am, so I'm a historian and philosopher of biology. Good afternoon from a very sunny, absolutely glorious afternoon, about 30 kilometers outside of Brussels in Belgium. So I'm at the Université Catholique de Louvain and in the Collegium of the Académie Royale de Belgique here. So a phenomenon that's probably familiar to many of you. I was really hoping that I'd be presenting to you a complete draft today, but it turns out that you're getting very cutting-edge preliminary data. This has been a very live and ongoing project for our group, trying to look at the nature of scientific disagreement, in particular in the context of taxonomy and biodiversity, and doing that both from a kind of old-fashioned close reading approach, but putting that in dialogue with some text analysis type approaches. So what I want to do today, I'll try to just very briefly motivate that idea. So why do you care? Why should you be interested in questions about disagreement, conceptual structure in the study of biodiversity and taxonomy more specifically? I'll run over a few ideas about what philosophers have thought we could do about this, how we might respond to cases of ambiguity and disagreement. But all that I hope to do pretty quickly, because I know who the audience is here. People are interested in getting into the nitty-gritty, the nuts and bolts. So I'll present some different empirical analyses, some different approaches that we've done on a corpus of articles in taxonomy, how we got that corpus, how we tried to validate that corpus, and then some various tools that we've thrown at it over the last 6 months or so, trying to mess with topic modeling, as well as some vector representation, some stylometric analyses to try to understand the cartography of this disagreement in this science. Close with a couple of future ideas about stuff that we haven't done yet that we know that we want to do. But the long and short of it is that I think what we've got here, this is why I thought of presenting this case at this project at a meeting like this, is a classic instance where there's a strong agreement, actually, about the presence of a certain empirical phenomenon, namely this disagreement among scientists, among practitioners, and that phenomenon is observed, is attested in both the biology literature and in the philosophy literature, and it's always done so as a rule in a kind of negative, worried sort of tone. If there really is a phenomenon like this in the literature, if it really is posing a problem, that's the kind of thing that's subject to empirical test and to empirical analysis. And so that was the idea that sort of motivated the project. And I think in that sense, it serves as a nice example of what's the kind of stuff that these tools can do for you. What kinds of philosophic or historical or scientific problems might you be confronted with that could push you to want to use these kinds of methods? Okay, so let me give you some background and context. So biodiversity and taxonomy. If you're not a philosopher of biology, although actually looking down the participants list, there's a strong representation of philosophers of biology in the room at the moment. But if you're not in the philosophy of biology, biodiversity is actually a really interesting concept. Obviously, if you're a person who lives in the 21st century, you're well aware that this concept is everywhere at this point. We spend a lot of time talking about biodiversity, threats to biodiversity, remediation of biodiversity, et cetera, et cetera. But if you actually try to unpack what biodiversity is supposed to mean in these kinds of contexts, it's actually pretty messy. I always make this joke when I present this talk to biologists that when they go home and, tell their parents that they work on biology or on biodiversity, like, what people have in mind when you hear the word biodiversity. It's like, oh yeah, it's this stuff. Things that look like that, or maybe in a slightly different environment, it's things that look like that, right? There's kind of a it when you see it approach to understanding biodiversity. And in part, this is because the concept's been kind of tense, if you will, from its very beginning. It was introduced to try to give a bit more value judgment side, a bit more of a normative edge to a descriptive, empirical, scientific, ecological concept. We know that right off the concept has to be sort of balanced between just saying that we want to care about or preserve single big charismatic important species like the whales or the panda bears or the polar bears or what have you, because we want to capture ecological relations and we want to capture species that nobody cares about. We want people to conserve insects too, right? But it's also got to be smaller than just everything, because you can't tell people as a matter of practical conservation policy that you need them to conserve literally all of life on Earth, because that's going to be a non-starter from the get-go. So you want to try to find this really subtle middle ground here, which makes for a lot of work for biologists. Famously in the biology community, this has turned into a sort of hunt for indicators of biodiversity. So biodiversity is a concept that's very much tracked by proxies. Perhaps you just count species. That's this measure of species richness. How many species are there in a given area? You maybe make some corrections to that number because you'd like not just a lot of very similar species, but a lot of very different species to capture a kind of intuitive sense of diversity. You might talk about traits or characters, take a kind of morphological approach instead of talking about species. You might talk about a kind of network approach, a kind of structural diversity of ecological communities, that this is one of the cool things that evolution made, is a bunch of structurally different answers to these questions. You might talk about diversity of ecological niches, so the kinds of things that organisms are doing, the kinds of lives that they live. That might be the kind of diversity that you want to save. And of course, now with widespread, extremely cheap, high throughput genetic sequencing, you might just talk about genetic diversity. So let's just see, we want as many different kinds of gene sequences as we can to exist on the Earth. That's all well and good, but of course, a lot of these involve talking about species, and any biodiversity studies that rely on species inventories, counting up species, are going to inherit the rampant uncertainty and disagreement found in taxonomy. So nobody agrees about what a species is, and even if you agree about what a species is, you often disagree about which species you should carve a particular collection of organisms up into. So this has even been called, here, this is an article in Nature, this has been called taxonomy anarchy. So this is, I mean, when right, you guys know this, when you're a philosopher of science and you see a journal article title like this, you're like, yes, this is where they need us. We're ready to go to work. What can we do about it? So what kinds of responses in the literature have there been to the presence of this disagreement? The first is a kind of fundamentalism, and the best description of it that I know of is in this paper by Beckett-Sterner and colleagues. To be very clear, Beckett's not arguing for this. He argues against this, but he expresses it very clearly. Where they note that especially in cases of database curation and ontology development, there's a very strong pressure, for obvious reasons of interoperability, toward grounding our classifications, including taxonomies, in a consensus about the definition of the entities that are being classified. And this is a very old way to think about the sort of response to ambiguity, right? I always feel like a proper philosopher when I can put an Aristotle slide in a talk. Here's Aristotle in the Rhetoric saying that just what speech is to communicate clearly. Speech which fails to communicate a plain meaning will fail to do just what speech has to do, right? So this is a very old idea about what language is for. So that might be a first idea. Let's eliminate this disagreement. Let's make everyone agree about one referent for these concepts. Second response might be to become a skeptic. One famous skeptic in this context is Sahotra Sarkar, who's just sort of said, well, okay, look, biodiversity is implicitly defined as what's being conserved by the practice of conservation biology. It's just whatever it is that you're conserving when you're doing conserving. And you can say some more fine-grained things than that, but at the end of the day, you should be skeptical as a result. Or Carlos Santana, in what is one of my favorite paper titles ever written in the discipline of philosophy, Save the Planet, Eliminate Biodiversity. Provocative paper, but I think a really important paper where he argues that, look, this is just kind of a catch-all word that we use to cover what it is that we really want to value in the natural world. So we should just ditch this and talk about what we actually care about instead. Okay, you might also take a values in science kind of approach to this problem. And here, this is work from a postdoc who's actually now in my lab here, Stein Koenigs, who's written some great stuff on this, to whom I owe a lot of what's coming up. I'll mention his name a few times more here in a bit. Who's actually tried to make the argument that looking at the value judgments in biodiversity and taxonomy will help us unpack this kind of thing. And this is also a very old kind of recognition. So this is here Soule, who was considered by many to be the founding figure of conservation biology, in one of the founding papers in conservation biology in 1985, writing that the conservation biology is a crisis discipline. It's like doing surgery or thinking about warfare. You're always working without all the facts. You're working in a mixture of science and art, and you need intuition as well as information. This is just a very explicit way to say, look, we're appealing to other kinds of background and normative commitments. Other folks on our project. I'm sorry. Let me say this very briefly. So one response to that kind of value judgment angle might be to say, okay, well, sure, maybe biodiversity or conservation science are value laden in that way, but surely taxonomy, which is the kind of raw material that you feed in to biodiversity and conservation efforts, that's value free. But an increasing number of figures, and here Stein's been on the front edge of this, have started to push us to thinking about whether taxonomy might just be as value laden as conservation biology. And so we've got some case studies under development, other parts of this project, allied groups working in Flanders that I'm not involved in. Here's an orchids example where they do a very close reading, trying to see the structure of these value commitments in the work from these practicing scientists. All of that briefly to sort of gesture at why you might think there's a problem here, what kinds of aspects you think that problem might take on, but also to try to give you a flavor that everyone from biologists to philosophers has this inkling in working in these fields that disagreement in biodiversity and taxonomy is a huge deal, is perhaps very seriously harming our conservation efforts, and at the very least is something that we need to have a theory about. And you can have that theory from a purely philosophical perspective, as these three examples that I've just quickly run over would indicate. But what I want to play with, right, in this project and in this talk today is to think about applying empirical tools to this. So the first thing that we did is we got our hands on around 40,000 articles in hardcore taxonomy. These are the kinds of journals that you go publish in when you want to present a new species. Like, this is where you go to describe the holotype, really talk about this is why you think this deserves to be promoted, this particular organism deserves to be promoted to the species rank, et cetera, et cetera. This is the practicing systematics. Temporally, let me just give you a couple of kind of characteristics of this corpus. So that was the journals. Temporally, this is also kind of unsurprising, but axis labels are a bit hard to read, but that graph takes off around 2000. Of course, it crashes around 2020 because of various issues about corpus construction and moving walls. Climbing to about 3,500 articles a year. Unsurprising, right? This is basically the graph. We saw a graph that looked almost exactly like this 2 or 3 times yesterday, because essentially this is the graph of the distribution of digital PDF format journal articles in global academia, more or less. So yeah, this is a pretty unsurprising graph. So we've got these 40,000 articles that we could get our hands on through various kinds of agreements, copyright negotiation, nonsense, etc. If anybody's interested in my rant about copyright, feel free to ask in the Q&A. I could go on, but I won't yet. You might have a question though here, right? Which is, okay, so now we have these articles that we sort of the best we could get our hands on. How might you validate a corpus like this? And this is a really interesting problem, right? Because there's no such thing as the kind of ground truth list of species in the world. If you wanted to ask the question like, does this corpus representatively sample the species on Earth? That's essentially an unanswerable question. That is to say, it's the work of taxonomists that give us the lists of species that we have. There is no way to externally verify them, to kind of step outside of taxonomy and look at for instance, numbers of species present on the planet. People try to estimate these numbers and they vary by 3 to 5 orders of magnitude in our experience. So not reliable enough estimates to be meaningful. One thing that you might do though, that we tried anyway, is you might look at sort of rough distribution around the tree of life. So now we're not asking the question because that question's inaccessible. Do our papers sample well from the existing species on the planet? But instead we could ask the question, do our papers sample well from the sort of discipline of taxonomy as a whole? So we can take some big consensus data sets like the Open Tree of Life, which is a collective that tries to take all of the papers in taxonomy and systematics and mash them into one giant rooted phylogenetic tree of all species on the planet. Here I've colored this just with some kind of big colloquial groups. So the gold there is bacteria, green is plants, yellow is fungi, pink is vertebrates, chordates, blue is mollusks and annelids, and orange is insects. If you're arthropods, if you didn't know, insect taxonomy is kind of the 500-pound gorilla in the room. And then you might go compare that to the collection of species names that are found in our corpus. When you do that, you get something like this. And you'll notice immediately a couple of pretty significant changes. First of all, the bacteria went away. That I expected. Microbial taxonomy is very weird, and it happens in its own special journals that we don't have access to. It's also so weird that it's not clear that we want it in our corpus for theoretical reasons. So the disappearance of the gold segment doesn't bother me. The yellow segment also went away. That is to say that fungi have disappeared. That's a bit strange. We have a few fungal journals, but apparently not very many. I've also since learned from practitioners that fungal taxonomy is very interesting because it's often done by amateurs. Taxonomy is one of the few disciplines left in contemporary science where motivated amateurs regularly present real, serious scientific results. And they publish them in things like, the Journal of the Mycology Club of Northern Germany that's published in some guy's basement. And I don't have access to those texts. So I'm missing a big chunk of fungi. Plants are a little underrepresented. Of course, chordates are overrepresented because everybody likes mammals. And the rest looks pretty much as I'd expect. So, I mean, I'm not super thrilled with it, but I'm not super upset with it either. It's decent representation. There's no massive flaws in the corpus. This is just broken out in a bit more detail by those by a slightly different set of colloquial categories. But you see that insects, non-insect arthropods, plants, other invertebrates, mollusks, fish, reptiles, birds, fungi, and mammals. That's the first however many bars that was in descending order there. Okay, cool. So I'm decently happy with the corpus that I have. How can we try to unpack disagreement in the context of this corpus? What kinds of tools can we apply? Preaching to the choir a bit, I know, but I'll still, on the off chance that there are folks who have never heard of topic modeling and that it wasn't already defined 6 times in the talks in the second half of yesterday that I wasn't able to attend. So I apologize in advance. It probably was. But a nice, very rapid way, anyway, to say what topic modeling is to describe it as a kind of unsupervised dimensionality reduction algorithm, right? So you might think of documents as being spots in a 172 million dimensional space. That's the number of types in our corpus. There are 172 million distinct words, distinct kinds of word. You might think of documents as being locations in that space, and you might think of topic modeling as a very fancy way to smash that down to a 125 dimensional space. And in particular, not just any 125 dimensional space, but the fancy fun thing about topic modeling is you tend to get spaces where the 125 dimensions, if you go interpret them, feel meaningful. That's what justifies calling it topic modeling. When you go look at the dimensions look like subjects that a document could really be about. It's a nice way to think about papers as being sort of a mix of different topics. So now, how do we interpret topics? Let me just give you some examples. Again, my apologies if somebody already basically did this yesterday afternoon. Here's an example of one that's fairly easy to interpret. Here's topic number 16. I can tell you by looking at correlations that it's popular in mammals. It's popular in people who study mammals. Mammal species names are mentioned very often. And it includes words such as Colombia, Peru, Venezuela, Ecuador, Panama, Brazil, Rica, Del, et cetera, et cetera. It's like that cat sneaks in at the very end as well there. Long story short, right? Okay, cool. We can understand this. These are the words that are picked out as being most probable in the topic. Those values, those numbers there are the probability that once if a document is using this topic, that word will be drawn from that topic. I can say more about the underlying model here in the Q&A if people are interested and don't already know. But if you look at the probable words here, that sort of seems like we're talking about Central and South American collection sites, right? Largely, especially for mammals. But in general, we're talking about the kind of science that you do when you go pick up specimens in Central and South America. So that's a good example. That's an example of what happens when topic model interpretation goes well. Let me give you a cautionary tale, right? Here's topic 31. And if you do a bunch of furious Googling, because you're me and you're not a lepidopteran biologist, but if you go do a bunch of furious Googling, first of all, you already see Lepidoptera and forewing and leg and scale, male, female, and genitalia. And so now I'm thinking, okay, Lepidoptera, that's the family of the moths and butterflies. So I'm thinking to myself, moth and butterfly anatomy, especially reproductive moth and butterfly anatomy. Cool. All right. That seems plausible enough. Now, I mentioned on the last slide that topic 16 seemed to correlate really well with mentioning mammal species. So now a priori, right, I read this list of probable words and I'm thinking to myself, well, this is going to correlate with insects, right? Because this is an insect topic. Except that actually there is one really high, really clean correlation, and it's with papers that mention mollusks. And there's a reason for this. If you back up to this list of words, in addition to things like male genitalia species and female, down there about halfway down, you see bursae. Bursae is just the anatomical word for a sac, anything that looks like a sac. The problem is there's too many burses. The mollusks have lots of burses too. And the topic model, because it doesn't preserve sentence structure, it just looks at documents as sort of positions in this space of word types. It doesn't know when bursas, at this resolution, of course, you could make a topic model with more topics, but at this resolution, it doesn't know that those bursas aren't butterfly bursas, that those bursas are, in fact, mollusk bursas. And so it's collapsing this down. And if you're not very careful when you're looking at interpretation on these topics, you're going to kind of run into some trouble. A couple of just interesting topics that I just wanted to mention, because if you're thinking about characterizing a corpus, this is the kind of thing that topic models are really good at helping you do. Topic 9, which is a very popular topic, except in non-insect arthropods, fish, and fungi, feels like it's just words about traditional specimen collection. right? So species, which is popular in lots of these papers for topics for obvious reasons, but then forest, habitat, area, collect, cite, record, range, specimen, region, number, sample, distribution, right? Okay, that kind of thing is very popular, again, except in non-insect arthropods and fish, because they live in the water. So these are the kinds of terms about terrestrial specimen collection, and also not for fungi, which I don't have a slide to show you about this, but because fungal specimen collection, it's actually a different topic that talks about being on the ground in a forest. It includes things like floor and soil and litter, because you crawl around on the ground when you pick up fungal specimens. And those words are missing in this topic. And so that's really delightful. That starts to give you a hint that, like, the topic model is really telling you, it's really grabbing on to something about the content, about the semantics of these papers. Similarly, there's one topic, and I'm a little sad about this because it makes it a little hard to distinguish hypotheses about what's going on in this topic, but there's one topic that lumps together everything about molecular phylogenetics. So if you use sequences or analysis or molecular or DNA or clades or genes or genetic or CO1, which is a gene that's very often used for DNA barcoding, or datum or base, et cetera, right? This is, if you're doing molecular work at all, your paper is dipping into this topic. And that is among the top 20 most significant probabilities for any topic of any variety in a bunch of groups. It's top 5% everywhere. It's radically the most common topic in the corpus. So everybody is using molecular methods. And that actually, a priori, was a little bit surprising. We actually expected to be able to do a little bit more fine-grained distinguishing between who's more interested in molecular tools and who's more interested in fine-grained morphological tools. And to first order, the answer is essentially no one's not using molecular biology anymore. It's all molecular all the way down. Okay, how about disagreement? So how can we use these kinds of methods to start to get a handle on the disagreement question? And the way we started with this is, again, Stein Kunix, the postdoctoral fellow on the project, sat with a bunch of papers, a collection of, I think, a dozen articles that he knew were representative of the kinds of papers in which taxonomists fight with each other about stuff. So these are papers where disagreement is going on. And he drew out lists of words that seem to be repeatedly invoked in the context of those kinds of disagreements. So for instance, here's the disagreement list. There are 4 categories I'll show you in just a second, but here's the disagreement list that includes words like critique and doubt and opinion and disagree, reject, rebuttal, misunderstanding, misconception, misclassify, obsolete, et cetera, right? The kinds of words that you would use to very explicitly write about how you were disagreeing with the current state of the journal literature. And he put together 4 lists like that, and then I validated them into the corpus to see which forms of which words were really used by the articles that we had. One for terms that refer to sort of invocations of epistemic values, so talking about bias or accuracy or precision, et cetera, et cetera. When do scientists really talk that way? One that you just saw for disagreement, one for just pejorative evaluations, so talking about people doing bad science, essentially. And then a more general list for sort of the kinds of words that you would use just to talk about changing taxonomy in a much more neutral sense, about revising or reclassifying or promoting species to a different rank, et cetera, et cetera, et cetera. Just more, less evaluative, more descriptive discussions of taxonomic change. And so now what can you do? Well, you can ask the topic model. So what topics are likely to select words from our lists of disagreement terms or similar kinds of words? I'm going to drop off the taxonomic change list for the rest of the talk, actually, because it's much more general and analyzing it is much harder. So that's actually a project for future work. I'm really going to lean into the disagreement stuff for what's left of the talk. Disagreement terms are super popular in topic 43, epistemic value terms in 91, and pejorative terms in 43 and 120. What does that actually mean? Well, let's unpack these. Topic 43, popular both in the disagreement list and in the pejorative evaluations list. Now, this includes things like species name and description, but then also if you see publish, nomenclature, code, international and zoological. That's the code that writes down the rules for how to present a new species, as well as type, original, valid. So that feels like the kinds of words you would use to introduce a novel species in the literature. And you also very importantly see the word synonym here. I'm going to come back to this. But declaring that two species are in fact synonymous is the opposite movement of introducing a species, right? That's deleting a species from the literature. Hey, there's this paper. It says that X is a species. You're wrong. Actually, X is a synonym of Y. So these are the sort of addition and deletion terms from the literature. And it's really interesting that both heavy disagreement terms are very popular in that topic, relatively speaking, as well as being insulting is very popular in that topic. Topic 120 that was only popular on the pejorative list. Notably, this is really interesting. This is the only topic that includes words for taxonomic categories higher than species. So you see the words genera, genus, and family. Those words are not in the most probable words for any other topic. And you also see analysis, relationship, phylogeny, clade, classification, right? Plausibly enough, this is people arguing about how to rank clades. So is this a species? Is it a genus? Is it a family? Where does it go in the hierarchy? How should the hierarchy be structured? What's the correct subsets, et cetera? And this is really another very popular place to be pejorative at one another. So another really interesting site for disagreement. It wouldn't be a real digital humanities talk if I didn't present one slide that contained data that I have no idea how to explain or what it means. So this is my slide where I show you data that makes no sense to me. There's the one topic in which radically epistemic value terms are much more popular is about the anatomy of decapod crustaceans. So I am still not really sure what's going on here. My only cautious hypothesis is that the model is getting confused by the word robust, which you see over there on the far right. I need to find a crab physiologist to talk to me about how you describe crabs in journal articles. But yeah, I don't get this one yet. I so don't get this that I actually had to add support to my slides package for putting emojis in slides so that I could just put the thinky face. I don't know what's going on here. Now, you might be looking for more precision, right? And I especially feel the need for this in the context of things like topic 43, which had both notably the terms for introducing new species and terms for eliminating new species. And of course, I could just train much bigger topic models and try to see if I could break apart those topics that seem to be lumping things together. But another thing that you may know if you worked with topic models, this was already a 125 topic model over 172 million types. Doing a topic model that was double or triple or quadruple this size starts to get really computationally problematic. So I'm worried that I'm not going to be able to do that. It was also a nice, I think, a case study or a time to try out some novel methodology in our group anyway. So what we wanted to do was look at document vectors. So this is, if you've played with Word2Vec online, Word2Vec is now fairly old, as is in fact Doc2Vec. But the idea is that you build, much as you did with topic modeling, you build vector spaces, lower dimensional vector spaces, in this case a 100-dimensional vector space. Trained directly via machine learning in a radically different way to the way that, in fact, it's a neural network training system instead of an LDA training system. So it's a radically different training system with regard to how that vector space is inferred compared to topic modeling. But a long story short, you represent words as vectors in that space, and then via another algorithm that was developed at Google in the mid 2010s, you represent documents as well as vectors that occur inside that same space. So everything lives in a common basis, both words and documents. And the idea is that the directions of these vectors are taken to capture something like where they point in semantic space. a sort of meaning space. If you've ever played, there's some lovely ways to play with Word2Vec online. One of the nice things you can do with it is you can ask it via vector geometry to find, for instance, the word that has the opposite meaning to another word. And if you put in the word king and you click opposite, it says queen. Like, it gives you some of the kind of semantic opposites that you might expect in a very impressive kind of way. And you can visualize that. This is a bit, you've seen UMAP yesterday in Max's talk, but you can visualize exactly this kind of thing. This is just a clustered UMAP of a vector model, document vector model for all 44,000 documents in our corpus. Delightfully, I could show you an interactive version of this as well, but it clusters pretty well. And if you go mouse over these clusters and start to unpack them, you find, which is very reassuring, that the clusters tend to be organisms of interest. So, like, I remember this pale green cluster in the top middle there is spiders, for instance. That's all spider papers. So that's nice. That's also kind of reassuring that the model is grabbing on to genuine significant semantic differences in the papers. So then what we can do, right, is we can take each of those lists of disagreement terms, and in the same kind of way that Doc2Vec does, we can represent each one as a vector in this space, and we can ask, what are the documents that are closest to those vectors? So we did that for each of those lists. We took the 5,000 documents that are closest to the vector and the 5,000 documents that were farthest away from the vector. The first thing that we see is actually largely, so here the close documents are in this really, really bright green, and the distant documents are in this bright yellow. And the first thing that you see is actually, which I actually kind of wanted, there's no real clustering pattern here. They're fairly evenly distributed across the entire graph. I sort of expected this, because I wouldn't expect disagreeing or not to be radically correlated with what kind of organism you work on, right? I would expect people who work on almost everything to disagree at least sometimes. There might be very subtle trends, and I'm going to come back and talk about one of those in a second, but they're going to be more subtle. They're not going to be visible on a graph like this. So actually seeing this graph that doesn't really say anything was reassuring in a paradoxical sort of way. So now that I have those documents, right, I have a 5,000 document sort of positive for disagreement set and a 5,000 document negative for disagreement set, I can start to analyze them. So I can ask, for instance, which taxa are you more likely to discuss in papers that are in the disagreement set versus not in the disagreement set? So I pulled out all species names from the top and bottom 5,000 documents. There's a lovely package for this called GN Finder. And I compared relative risk, right? Just descriptive statistics, right? What's the ratio of the proportions, if you will, the ratio of the ratios of papers that mention a given taxon in the positive corpus versus in the negative corpus. And you find that there's more disagreement about mammals, almost 4 times more disagreement about mammals in the positive corpus versus in the, 4 times more mentions of mammal species in the positive corpus than in the negative corpus is the correct way to say that. Thrice as many mentions of birds, thrice as many mentions of fungi, and twice as many mentions of fish. I took the kind of standard clinical relevance threshold of 2 or 0.5 for relative risk to be meaningful. There's less disagreement about insects. So you only see about half as many insect species in the positive articles as in the negative articles. Already, this is really sociologically interesting in terms of trying to figure out which taxonomists are angry, what are they angry about, and why are they mad. Just this one slide is already really cool. I then thought, well, okay, what else can we do if we have these 2 corpora? And you might ask yourself, well, okay, other than the disagreement words, what terms do these papers use? So what words, for instance, pick out as marking out the positive disagreement corpus from the negative disagreement corpus. And there are a number of really nice algorithms for this. There's a lot of this kind of work in the literature, in particular in stylometry, so computational stylometry, where literature scholars are really interested in asking questions like, what words pick out, I don't know, late Henry James books versus early Henry James books. You can use that, actually, it has been used to test the hypothesis that James was getting dementia as his career rolled on. And in fact, the language in his late books becomes simpler than the language in his early books. Seems to be borne out by this kind of analysis. So I stole one of these algorithms. Technically, it's the Craig Zeta algorithm that's been developed over a number of years. Now it's a very old algorithm in computational stylometry, which gives you a list of words that are sort of indicative of membership in the positive or in the negative corpus. And in the positive for disagreement corpus, you already see some really interesting stuff. So you see appear, consider, probably, suggest, et cetera, et cetera. You also see synonym and synonymy. So notably, that worry that I had about pulling apart introducing species from deleting species seems to be readily handled by the vector models. Because you have positive, very positive disagreement correlation with synonym and synonymy, and a negative disagreement correlation with the words holotype, introduction. I think maybe you can see my mouse. Holotype, introduction, new, and NOV, which is actually the abbreviation for new in Latin that you use when you write down a new species word. You give it a name and then you say SP, NOV, new species. So right off, it starts to feel as though we've pulled apart introducing species and deleting species, and all the disagreement is in the papers that delete species. They're not in the introduction papers. The introduction papers all show up on the non-disagreement side. Probably some of you have already noticed the other shocking and really interesting fact about this, which is that on the positive disagreement side, you have Western and European, and on the negative disagreement side, you have China, and just off the end of this list, you have India. And as well, sociologically, this is extremely interesting. Western scholars are just much more disagreeable than Eastern scholars, it seems. And we have some preliminary hypotheses about this. On the one hand, part of this is assuredly due to the undeniable fact that there's just been less taxonomy done in places like India and China. And so they are, for instance, still busy just figuring out what species are present in their regions in a way that we're not doing in Western Europe anymore, where we've been doing taxonomy for 300 years in a fairly uninterrupted tradition. So that may be part of this story, but there may be more and more interesting other explanations of what's going on here as well. We're really interested in running this down. It echoes a few other things that we've seen in other parts of our project. So yeah, this is the kind of stuff that we have been up to lately. I mean, I think I can't give you our final, if you will, a fully packaged analysis of how I think disagreement works in the taxonomy literature. We're still up to our necks in this data, trying to figure out exactly what all this story tells us. But one thing I think is extremely clear is that, in fact, the patterns of disagreement in the literature are highly variegated. They depend on a lot of factors. They seem to depend at least moderately on what organisms you care about. They seem to depend at least moderately on where you work, on what kinds of whether that's where you collect your specimens or where your university is. All of these features are, I think, significantly more complex than we have given them credit for in the past. Coming soon to a data set near you is geocoding. Another obvious thing, especially confronted with these invocations of place names, would be to look up essentially proper place names in every single one of these papers and see how that correlates with the other kinds of things that are going on that I've already described in the corpus. If you've done geocoding, you know that's not an easy proposition. As I know from personal experience, if you write Google's research arm and you tell them that you have 500,000 locations and you would like to geocode them, you get back a very cold letter that says, "We know what you're trying to do. You're trying to reverse engineer our geocoder, and we will not allow it." So they don't like that. they're not real interested in hearing from me about geocoding the entire data set. So I'm gonna have to think about different methods for approaching doing mapping and performing GIS in these contexts. But in general, I think we're very close to having a much more nuanced and complex understanding of what is a really important ingredient in figuring out how should we respond to this situation of disagreement in taxonomy and biodiversity studies. And I think, at least for my money, when I started doing this work now 15 years ago, this is exactly the kind of stuff that I hoped I would be able to do with these kinds of tools. And I'm really excited to be able to be in the position of I think shining a lot of light on a really philosophically, scientifically, and socially interesting problem. So with that, really excited to take any and all questions, and feel free to get in touch. Again, huge thanks to Stein Konix, the postdoc in my lab on the project, as well as Max Bautista, who is the graduate student in the lab on the project as well. They've been super helpful. This has been a whole lot of fun for the last year and a half. So thanks very much.
Thank you, Charles. Thank you very much. We have several questions. I see Stefan.
Oh, thanks for that great talk, Charles. That was so thought-provoking and exciting. One small technical question and maybe a bigger technical question. So you mentioned pejorative language, but I didn't see it in any of your slides. So maybe you can tell us what that looked like. But I think my other question was, I'm just trying to think of ways you could ground truth whether you're actually tracking disagreement. Good. And, I have a hunch that there are just well-known disagreements in taxonomy. I think the lichen literature is actually an area where there's a lot of it. And so I'm just thinking that, how would you do that, right? Suppose you had independent evidence that here's an area, a set of species or something that there's a lot of disagreement about. What would you do to check whether you're getting things right?
Yeah, good. Those are two great questions. So yeah, first, just very quickly, the kinds of things that are on the pejorative list. Let me pick some at random. It's things like simplistic, exaggerate, harmful, dogmatic, subjective, confounded, trivial, unconvincing, excessive, misleading. It's that kind of stuff. Yeah.
Wow.
Another thing that I think is actually also important to say, I didn't actually say this out loud in the talk, but one thing that we've also found is those top 5,000 documents in the epistemic value list, the disagreement list, and the pejorative language list very profoundly overlap. So those top 5,000 documents only have around 6,000 unique documents in them. So the model is putting epistemic value judgments, disagreement, and insults in very similar regions of semantic space, which is already kind of interesting. Ground truthing, yeah, no, I'm absolutely with you on this, and I think I haven't thought through how I really want to do this yet. But the kind of thing that I do think we need to do, and it's the reason that I kind of gesticulated at this paper that so. I'm an FNRS project, which is to say that I'm in Francophone Belgium, but there is an allied project with the FWO, which is the Flemish-speaking part of Belgium that has a separate funding agency, and they're working over there doing very similar stuff, and we're very closely integrated projects. On the Flemish side, across the linguistic frontier from here, they are doing this, they did this case study in the orchids, and they really sat down and unpacked a bunch of very detailed papers about orchid taxonomy, trying to read in to exactly where are the fights, what are the fights, when are people getting, what is it that gets people worked up exactly. There's less disagreement than it actually seemed like there was once you unpack the literature, which was kind of interesting. I think we're going to have to pick a couple cases to do exactly that kind of work, because I think it's very important that we believe the results of these models. And be sure that the more kind of discovery-based stuff like this we do, the more worried I might be that we're chasing spurious correlations, and I want to be very sure that I'm not. And I think good old-fashioned close reading is the play, honestly. And yeah, actually, I'm gonna go see if we've got lichens. I'd forgotten about the lichen literature. Yeah, those people really get into it. That's a great idea.
Thank you, Charles. Guillermo?
Yeah, thank you, Charles, for that very good and inspiring lecture. I have several questions and comments, and perhaps the most important one that I have here is that whether you have considered to go in the direction of history. I mean, you have your papers, you have the label, the year of the publication of the paper, and then to consider the evolution of all that what you have done, but now adding the temporal dimension, and see, for example, you mentioned that in some papers, I think it's in the majority of them, you always see molecular DNA sequence and things like that. But I think if you go back in time, that was, of course, not always the case.
Yeah. Good.
Then you can also explore the introduction of technologies, and also, you can also add the political dimension to see how that has influenced the kind of space you are exploring. That's my comment.
Right. No, excellent comment and very well taken. In fact, it is sitting on my to-do list to do exactly that with that molecular topic, because it's very interesting that that topic is uniformly so highly probable, because that data set should go far back enough in time. I mean, in 2003, those methods were not cheap enough that everyone was using them, right? They were not in every lab yet. And so that we ought to see that. That said, one of the big challenges that we have wrestled with is, before the kind of development of these born digital journals that started to just magnetically attract all of the work in hardcore systematics, man, publication practices and systematics are crazy. And so tracking down a real historical corpus, I mean, I would love to, a bunch of my work is late 19th, early 20th. Like that's my bread and butter. I would love to be able to watch this over the long 20th century. Getting our hands on a corpus is really rough. It tended to be published in actual monographs. So, someone would decide that they were going to rewrite the monograph of the orchids. And oh man, that's going to pose all kinds of wild analytic challenges. And so I don't know how to do it yet. I would love to be able to, but I worry that, man, getting much back before 2000 is going to be technically a real mess, unfortunately.
Yeah, right.
Thank you. Arno?
Yes, can you hear me? Yeah.
Yes.
Yep. Great. Thank you very much. It was a super interesting talk, especially that you decided also to go beyond topic modeling and include these document vectors. And I have actually 4 questions I have for you. The first one is very technical. It's just a question: how did you get the plain text out of the papers? I assume you might have them in PDF, so that's a problem we are struggling with in our project. Second question: I didn't quite understand how you formed how you constructed this disagreement vector. Was it like, did you just kind of write all the disagreement words in one sentence and then that's the vector? Yeah, okay. And then the third question is, how did you get the document vectors? Was it the whole document, or did you take each sentence or each paragraph? And the question is, if you take more than one sentence for a document vector, have you tried using section vectors? Let's say particular sections in papers might be more these sections where you talk about the other literature and maybe talk more about why you disagree with the literature or you agree with the literature. So maybe these type of sections in a paper might be more valuable to compare against. And then something more like to go beyond that thing is, have you thought of ways to use additionally bibliometric indicators or measures, something like who cites whom, or some sort of citation, co-citation clusters, etc.? And yeah, thanks. That's all.
Awesome. 4 great questions. Cool. So how plain text? Fairly simple answer. Almost all of these papers, delightfully enough, had proper, reasonably accurate plain text layers in their PDFs. So they were actually digitally produced. In the few that didn't, the answer is Tesseract. So that's the best I've got for now. Obviously, that's going to have introduced error. Obviously, in 44,000 papers with 170 million tokens, we did no manual cleaning whatsoever because I do not have that much money. So yeah, that's the first question. How the disagreement vector, I'll answer this in combination with the next one. So we did just treat that list of disagreement words as a document and inferred it in the model once the model was trained. Which relates to the question of so how document vectors. The Doc2Vec paper is really interesting. The way that the Doc2Vec model works is, so obviously you can just compute the mean vector of every word in a document. Like that actually works okay, historically speaking, but it's not as good, it turns out, at least according to the guys who wrote the Doc2Vec paper. Let me be clear, like I am not actually a machine learning guy. But it turns out that you get better looking data, you get better looking results if you simultaneously train vectors for words and vectors for sliding 20-word windows over your entire document, or at least 20 is the window size that I used. You can pick your own window size. So you explode the size of your neural network. Obviously, it makes your training go much slower. But to be perfectly honest, the training on these models is shockingly fast. So that for those 40,000 document models, I train a vector model over my entire corpus in about 17 minutes on the i9 in my office. So it's shockingly efficient. It's orders of magnitude more efficient than doing a big K LDA. A 125 topic model is a couple hours on this box, and a vector model is 13 to 17 minutes. So I was very impressed by the efficiency of the algorithms. What I do? Yeah, please.
Just one quick question. The document vectors, do they preserve word order? Do they somehow embed the word order?
No, not within the sliding windows. Within the sliding windows, it's a bag of words model.
And also for your disagreement vector.
Exactly, yeah. So the order of my disagreement list doesn't matter, which is good. Obviously, if there was an order dependent effect there, that would be really bad. But there isn't, at least unless I've radically misread the algorithm. There's no order dependent effect. Section vectors would be really cool. The problem is that there's too many different kinds of sections in these darn papers. I can't figure out how to cut them up. I would love to, but you've got everything from there's just too many different kinds of papers published in these different journals. So if I restricted myself to one journal, something like Zootaxa, which is really big, it's the majority of my corpus, I could cut myself to that and then think about doing that kind of smaller analysis. That's actually also the thing that prevents me from doing bibliometrics, which I would also love to do. I don't have clear data on citations. I could extract it, but I could only extract it if I were confident enough in bibliographic presentation that I was sure that it wasn't slipping out from under me while I was doing a reference matching. I've got a postdoc working on this. I'd be interested to see what he ever comes up with. It's been a long and arduous process of trying to do sort of how can you do reference matching if you don't have, say, Web of Science, which my university is also not rich enough to have Web of Science. So yeah, we're playing with this. I don't know where I'm going to end up. But in both cases, I think my data set isn't giving me access to what I would need.
Okay, thanks.
Thank you very much. And we have time for one more question, Christoph.
Yeah, thanks a lot, Charles. Great talk as usual, and good to see you here.
Good to see you.
I had a couple more technical questions because I mean, overall, it's an amazing piece of research that you're doing. When you first mapped the tree of life with the species, did you use some sort of a named entity recognition type of algorithm to extract the species from the documents, from the articles? So are there all the species mentioned in all the articles, meaning that sometimes, some species may be mentioned several times, or several species may be mentioned in the same article? So I was sort of wondering how you did the mapping here. Concerning the top2vec and doc2vec thing, you interpreted the clusters on the UMAP as phylogenetically meaningful clusters. But the thing is, I was a bit surprised because Top2Vec will, cluster documents in the word space, but you may actually find documents that are related to one another based on different features, and typically based on the words that they use. And so in this respect, there is a very, you can use Top2Vec and Doc2Vec as a means to do some topic model. So I would expect, I would have expected a diversity of topics somehow being present as the clusters. So the clusters could be interpreted as topics. And so I would have expected, for instance, if you had a very strong phylogenetic cluster that you topic that you identify with the LDA, I would have expected that you would have found a very big cluster of documents in the Doc2Vec that would be just about phylogenetics, right, and not about species. So I was sort of wondering why you interpreted the clusters in the Doc2Vec as phylogenetic groups and not as topics, because you could actually compute the a synthetic vector representative of each of the cluster, and then I'd interpret this synthetic vector as representative of a topic. And then my last question would have would actually be quite simple. It's about, since you're interested in the present specific terms, disagreement. So you made a list of all these terms. How about just simply looking at the frequency of these terms, and then looking at the documents in which these terms are the most frequent? You could do concordance type of analysis, look at pairs of these terms being present together if you're interested in that. But you could also, just sample the document space just by looking at the document in which the terms are most frequent. And then you could also correlate that with a topic modeling, having the topic profile of all the documents. So. And then I would have plenty of other questions, but that would be enough for today.
Cool. Yeah. No, 3 good ones. For the first one. So yes, I'm actually just very lucky that the bioinformaticians solved this problem already. So there is a phenomenal GitHub package known as GN Finder that combines rule-based extraction with trained Bayesian classifiers with matching to all of the online databases of known species names, and does shockingly quick, shockingly high quality extraction of taxonomic names, and not just extraction, but then verification against those external databases. And it does things like it gives you the whole taxonomic path to the species. So you don't just get the species name, you can, which I actually need, right? I need to know if I want to just say, where are the mammals, right? I've got to go back up the chain from the species name and figure out whether something is a mammal or not, which is not inherently obvious, right? Given to you by this tool. Super great. Has saved me. I don't even know what I would have done if I didn't have that. So first question is that. Second question, I should be clear. I was being a little rough and ready. So I think the topics the clusters in the UMAP, I have a couple things to say about them. So first, I should be clear, largely but not completely phylogenetic. So most of the radically distinct clusters that are sort of far flung from the center, upon inspection, it's all the same taxonomic group. That's actually also a feature that's duplicated largely in the topic model. If you look at most probable documents, as you come to the center, things get weird, which actually is often the case with these kinds of UMAP representations, right? The things that show up in the middle are the stuff. You might have recalled there was a giant red cluster in the center of that image. UMAP doesn't really know what to do with it. It doesn't have any obvious distinguishing features. It's just kind of a place where the representation breaks down. For that reason, I should also add, I'm not putting any faith in the analysis of those clusters. I have not sat down and tried to really identify them. I don't even know what their numbers are. I actually don't even know, because I really did it just to visualize. I don't even know how many clusters I have. I really did it just to sort of see whether it looked like there were radically distinct clusterings. And then, of course, visually, that's the case. And a Louvain clustering, which, of course, I'm contractually obligated to use as my only clustering method, confirms that. Right. So yeah, no deep faith in clusters, not even really going to analyze the clusters further. It was largely because I would have been really worried if all of those terms were sitting inside of one cluster, disagreement terms or non-disagreement terms, because that, to me, it's not as though everyone who fights in taxonomy only fights about in a very limited part of the literature, right? Disagreement should be a more broadly distributed phenomenon than that. That was the only intuition that I was looking to kind of solidify with the clustering. Why not just search? I mean, fair question, right? And I did do a little bit of just searching before turning to the vector models. But the truth of the matter, I mean, what it comes down to is I spent a long time trying to think about and looking at various algorithms for. So if the question that you would like to ask is which documents contain relatively proportionally more of this list of words than others, I'm actually not sure that the kind of a priori best way to answer that question isn't with some kind of vector model representation. Because I don't just, it's not even absolute frequency, right? It's like an overrepresentation of the stuff in the list versus the stuff that's not in the list. And that's a very hard thing to try to quantify if you're just looking at a frequency table. And especially if you're in a case where I think, a concordance is going to have so many too many entries for me to really be able to wrestle with in detail. This is also where I think we can close full circle. This is where you get back to Stefan's question as well about probably this is where you need to come in and also be doing some close readings to really see the papers that look extremely disagreeable, for lack of a better way to put it. How do they read? Right? What are they really doing? And I got to get to work with some of the other members in my project team who know a whole lot more taxonomy than me. I know a little bit of taxonomy, but not enough to do that kind of close reading work.
Thank you, everyone, for your wonderful questions. Thank you, Charles, for this very illuminating talk.